Hello, everyone. And welcome. So today we're going to be covering my personal favorite topic of this semester, and that's going to be bacteria, archaea, and viruses. So the microscopic organisms. Now, microbiology has a pretty rich history. It really dates back to about the 1500s. It started with a Italian physician named Girolamo Francastaro. I think I said that right. Francastoro. There we go. Um, and it dates back that long, which is surprising on one hand, but not too surprising on the other. Microscopic organisms, as their name implies, are very, very small, but they have played an integral role in shaping human population. And this has been true not just today with as we're dealing with COVID, but has been true throughout the history. And so you can think of all the great pandemics, thinking of things like cholera or measles, or even modern ones like HIV, or even other ones like the Black Plague. Microorganisms and humans have basically been hand in hand since day one, and they've acted to dramatically shape our population. Now, this Italian physician in 1840s, suggested that disease was caused by unseen organisms. Now, this is in direct contrast to what most other people were believing at the time, in which disease was caused by what was termed miasma, or simply bad air. Now, as we know, disease can be carried by air, but this physician, he basically said, nope, it's not just the air, it's microscopic organisms inside the air. So he really got us started thinking it, which is I think a pretty cool thing. But microbiology as a field didn't really take off till about the 1700s with Antony von Leeuwenhoek. Now, von Leeuwenhoek, you might have heard his name before. He's the inventor of the modern day microscope. So anything you see under a microscope, you really owe to Leeuwenhoek. He developed really, really precise microscopes that allowed him to describe microscopic life for the first time. And Leeuwenhoek has these really cool experiments where he would scrape his teeth and get all the gross bacteria on his teeth, look at them under his microscopes, and see all the microbes moving. And then he would drink scalding hot coffee because why wouldn't you drink scalding hot coffee? And then he would do his teeth scraping again, and he would actually see that the microbes were no longer moving. So he did this really cool early sterilization experiment, but again, all made possible by his microscopes. Now, Leeuwenhoek is a really interesting guy. He was kind of, for lack of a better word, a jerk, and he actually was so paranoid about his microscopes that he actually hid his technology away, and actually single-handedly stalled the field of microbiology for about 100 years, simply because he wouldn't share anything of his materials, and he actually destroyed all his microscopes when before he was about to die. So his technology was lost for about 100 years or so. So again, kind of a jerk, single-handedly stunted the field of microbiology for a wee bit of time. Fast forward about 100 years after Leeuwenhoek, we have Louis Pasteur or Louis Pasteur. Now, Louis Pasteur is an interesting guy. Now, you might recognize his last name, Pasteur, as in pasteurization. So you guys might have drinking pasteurized milk or pasteurized cheese, and you owe that to Louis Pasteur, who developed the process of pasteurization to essentially heat kill microorganisms in dairy products and other aspects of our food chain. So Louis Pasteur, pretty important guy in microbiology. He also had a couple really influential people that worked for him. So, for instance, one of his students, not students, I should say, one of his research assistants, um, was his name was Petrie. And when you go into a lab and you work in a lab, you commonly use what are called Petri dishes, which again is from a lab researcher from Louis Pasteur's own lab. So, kind of an important figure in microbiology. Now, Pasteur also came forth with the idea that's refuted this idea of spontaneous generation. Then spontaneous generation basically says that living things are spontaneous or rise spontaneously from other living things. So like, if I have a flower, it just spontaneously comes out of another flower. And that's not quite how life works. But Pasteur really stamped that down and said life has to rise from other similar types of life. And he did this with microscopic organisms. Now, not too long after our friend Pasteur, Robert Koch or Koch or Robert Koch, there we go, Robert Koch. He was a scientist, a microbiologist that worked on anthrax. And you guys might remember, or maybe you're too young to remember, but the anthrax scare during the sort of the 9/11 years, September 11, 2001. people sending it as bioweapons through the Postal Service and things like that. But anthrax is a normal human pathogen. It's found on rotting organisms, so it's not too uncommon. But studying anthrax, uh, Koch proposed four what we call Koch's postulates to prove a causal relationship between a microscopic organism and his disease. And so what Robert Koch told us is that first, that microorganisms must be present in every case of the disease and absent from healthy individuals. That putative causative agent must be isolated and then grown in a pure culture. That same disease must result when the cultured microorganism, so from step 2 here, is then used to infect a healthy host, and then the same microorganism must be isolated again from the disease host. So let me give you a breakdown of how this would look. If we say, I'll use COVID as an example. So we have to see that all the people that are suffering from this severe respiratory illness all have the same virus. Then step 2 tells us we have to isolate the virus, grow it in a pure culture. Then we have to take that same disease, reinfect the host with it, and then re-isolate that microbe from the host. So that's Koch's postulates, a nice stepwise fashion by which we can confirm whether or not a microscopic organism causes a disease. And again, really important for modern medicine. And that's because, as I mentioned, our medical systems and human history are directly tied to microorganisms. So pretty cool guy. Now, that's some milestones in microbial history. There's lots more, right? The invention of penicillin by Alexander Fleming in the early 1930s, another big milestone of microbiology. You could think of technique that we're talking about by the gram stain developed in the 1800s. Again, another milestone in microbiology. And you can even think about a modern milestone, right? The Human Microbiome Project started in the early 2000s, where we tried to catalog all the microorganisms associated with healthy humans. And so microbes have a rich history, again, almost 400 or 500 years now, and really are directly tied to us. Now, we are so interested in microorganisms for this course because well, they're kind of everywhere, right? From the top of the atmosphere down to the bottom of the ocean, you find microbial diversity everywhere. And it underpins every aspect of every piece of life on this planet. And we might not like to think of the idea of this, but you as an individual, you have about 30 trillion cells as an average everyday adult. As an average everyday adult, you have about 38 trillion microbes living on and inside you. So even you, as a basic living unit, are subject to microbes, and you could not live without your microbes. And that's not even considering all the other things, like we need microbes to grow our food, and we need microbes to keep our ecosystems healthy, and so on and so forth. So microbes, super diverse, and underpin every aspect of the course we're going to talk about. But we're going to devote today's lecture just to talking about our bacteria, our archaea, and then a little bit about our viruses. Now, prokaryotes, as we discussed in the Origins of Life class, encompass both bacteria and archaea, and they're the oldest, structurally most simple, and the most abundant forms of life. And I say that they're most abundant, and I gave you that fact of 38 trillion microbes live on and inside you. That's just a kind of a drop in the bucket in terms of how many microbes are on this planet. We know that there are approximately 10 to the 30, or 10 followed by 30 zeros, of prokaryotes on this planet as of today. So they constitute about half of all living biomass or living tissue on this planet. And again, it's all things you simply cannot see. So I hope I've made the case of how important and widespread and abundant these guys are. But remember, these organisms have lived on this planet since basically the beginning when life started, well before eukaryotic organisms appeared, and they've been evolving for that amount of time. And as we discussed, microorganisms can evolve pretty rapidly, right? As we saw with HIV, that can happen in a really rapid time frame. Now, microbes are the most abundant, they're also the most diverse organisms on the planet. Now, there are currently about 15 to 16,000 described species of microscopic organisms on this planet. That is a drop in the bucket. We estimate that there's probably closer to 100 million on this planet, but definitely at least 5 million on this planet. So really, really high amounts of diversity in both. Him in just a moment. So they're always pretty much unicellular. They're very, very small. Now, cell size does vary tremendously, but the vast majority of bacteria and archaea are less than 2 microns long. So, 2 micrometers, so really, really small. There are some that you can see with the naked eye, but the vast majority of prokaryotes are unseeable, less than 1 to 2 microns in length. So very, very tiny. And I just put that in perspective, right? 1 micron in diameter, a human cell is about 100 microns in diameter. So 100 times smaller than your average human cell. Now, they don't have any membrane-bound organelles, right? So they don't have a mitochondria or a nucleus, but they do keep their DNA in what we call the nucleoid region. Now, they have, unlike humans, just one single circular chromosome. It's double-stranded DNA. So no organelles, everything in terms of their genomic information is found in that nucleoid region. So just hanging around in the cytoplasm. Now, as you may recall, humans, our cells reproduce by mitosis. Bacteria do not undergo mitosis. They divide by binary fission. So this is just a nice little picture of what we call a biofilm. Now, as I mentioned, most microscopic organisms are single-celled, and you can see that here. Like, for instance, there's single cell here, single cell here, here. But you notice that they're kind of growing on top of one another, forming this 3D structure on top of this cylindrical object. So pretty cool. So this is what we call a biofilm. And if you've ever, gone, say woken up really early and you didn't have time to brush your teeth in the morning, and then you get to the end of the day, your teeth are all grimy and gross and nasty, and you feel kind of gross, right? They have like this film on top of them. Well, that film is a biofilm of bacteria. And this is what a biofilm looks like when you zoom in at about 750 times magnification. But again, a bunch of single-celled organisms living on top of one another and living together. Now, prokaryotes also have relatively high genetic recombination, as we've discussed in our genomic evolution class, lots of horizontal gene transfer, but no sexual reproduction at all. As I mentioned, there's no membrane-bound organelles or anything like that. Nothing crazy. A lot of bacteria and archaea have what we call flagella. So they're just basically whip-like tails that propel them through their environment. And they have very large amounts of metabolic diversity. So whereas our cells can only do, regular respiration and a little bit of fermentation, bacteria have evolved a wide range of metabolic pathways that allow them to basically live everywhere and anywhere on the planet. Now, I've been broadly grouping bacteria and archaea together as prokaryotes, but I will say that bacteria and archaea do differ quite substantially, right? All those characteristics that I just pointed out, they all share, but they do differ in 4 key ways, which includes the structure of their plasmid membrane, their cell walls, how they replicate their DNA, and how they express their DNA. Now, very much like us, bacteria and archaea have a plasma membrane. Now, bacteria and archaea's membranes are distinct, and I'll show you what that looks like in a sec. But archaea membranes are formed by uh, glycerol linked to hydrocarbons by ether linkages, whereas our cells and bacteria cells have ester linkages. And so let's look at what that looks like. Now, this is a bacterial or eukaryotic lipid, and you'll see the glycerol head here linked to fatty acids. And we have our ester bonds right here. COC, that's an ester. So that's our, I'm sorry, COCCO. So this area right here, this OCO here part, that's an ester. My bad. And so that's our bacteria and eukaryotes. And our archaea, as you see, they don't have that extra O. So this is just the ether bond. COC is characteristic of a chemical bond called an ether bond. So that's our differences. Now, eukaryotes and bacteria form lipid bilayers, but archaea also have this really interesting capacity to form what we call a monolayer. And so they have, again, it's linked by ether linkages. So we have that COC, right? That sort of linkage is here. But you'll notice that it forms a monolayer. So as opposed to 2 of these stacked on top of each other, we just have a single monolayer here. And again, only found in archaea. Bacteria and eukaryotes are limited to this. Now, the cell wall is not just an important part for plant cells. The cell wall is important for all prokaryotes. Now, bacteria and archaea have cell walls. Bacteria have what cell walls comprised of peptidoglycan, whereas archaea lack peptidoglycan. They have a compound called pseudomurin. which is not too different from peptidoglycan, but it is distinct enough. Now, DNA replication is a bit different in prokaryotes than it is from eukaryotes. However, archaeal DNA replication is much more similar to our way of replicating DNA than they are to bacteria. Simply just because of the waste or sort of the the proteins involved in the processes. And we're not going to get into it, but I just figured I'd make mention that DNA replication is different, as well as gene expression. Archaeal gene expression, as well as the translation of the mRNA products to proteins, are much more similar to ours than they are to bacteria. And that kind of makes sense when you start to break down the evolutionary histories. So when we looked at a similar phylogenetic tree to this in the origins of life class, we can put these three domains of life on a phylogeny, and you'll notice that they all share a universal common ancestor down here. But bacteria did split off relatively early from this ancestor, followed by archaea, and then you'll notice that the closest relative to archaea are actually eukaryotes. And again, we see those similarities reflected in DNA replication and gene expression as well. But again, three distinct classes of organisms. Now, you might be asking, where do viruses fall in? And the reality is we don't quite know where they fall into the schematic yet. They are so odd. They don't quite fit this three domain of life thing. So stay tuned on that. People are still actively working on the best ways to stick viruses into this into this wild and wacky tree of life. So when we talked about classifying birds or snakes or whatever we were discussing in evolutionary classes earlier in the semester, we were looking mostly at morphological traits. Right? We talked a little bit about DNA, but by and large, we were thinking mostly about morphological traits. Now, bacteria, we originally started classifying them based upon morphological traits, including their capacity to be photosynthetic, whether they're motile, whether they're unicellular or colony forming, or even filamentous, whether they could form spores, or whether they're human pathogens or not. So those are physical characteristics. And we can also throw in metabolism, right? Do they break down this type of carbon versus this type of carbon? So that's how we started classifying microbes early on. But in modern times, we've moved away from that a little bit. We still do it a lot, but we've moved away a little bit. And we started classifying microscopic organisms based upon their amino acid sequences of their key proteins. We've also been looking at what we call percent guanine and cytosine, so how much guanine and cytosine they have in their DNA. I'm not going to get into this too much, but it's a kind of interesting way to classify organisms. You can drop it into Google and you can learn some interesting things about how we classify organisms based upon what we call GC content. And there are other ways, including nucleic acid hybridization and whole genome sequencing. But one of the most common ways we actually classify bacteria is with this gene right here, the 16S ribosomal RNA gene. Now, ribosomal RNA, it's a structural component of the ribosome. So, oops. So it's found in all bacteria. It's also found in all descendants of prokaryotes, such as mitochondria and chloroplasts. And there's also a variant of this gene found in our own genome. So it's a pretty common gene, but it's something that's conserved and has we have a great understanding of how the gene evolved. So this is actually the gold standard for identifying bacteria and archaea nowadays, sequencing of the 16S ribosomal RNA gene. And again, it's just a gene that encodes for the ribosomal RNA that's associated with the small subunit of the prokaryotic ribosome. Now, outside of molecular characteristics, once we essentially classify organisms, whether it's via their metabolism, whether it's their genome, we can actually start to compare many of these things to what we call Berge's Manual of Systematic Bacteriology. It's a really robust book. It's like 900 pages of crazy detailed information about metabolic processes of microbes. But we can start to compare what we know about our microbes to what's in Berge's to better help identify our organisms. But the reality is we don't know most organisms, right? As I mentioned in a previous slide, 99% of all bacteria have not been identified yet. But the ones that we have identified, we actually broadly classify into some pretty major categories. Now, within the archaea, there are two main branches. This is the only branches found in archaea, by the way, but these are the two major ones. Now, we have the Euryarchaeota, which we associate with a couple different places. So we associate them with extreme environments, and we also associate them with the production of methane. So that's our Euryarchaeota. And then we have our Crenarchaeota, which are the most species diverse of all the archaea, and we associate with them basically everything: coastal oceans, sediments, um, soils, ponds, rivers, so on and so forth. Creniarchaeota are much more widespread than their counterpart, the Euryarchaeota, and these are just the two main branches of the archaea. There are, I think, if I'm not mistaken, about 25 other main branches of archaea that are not uh, mentioned here. Now, when we get into our bacteria, we can actually start to group them into a couple big classes. Now, we can start off with the thermophiles, which include a couple of bacteria called aquafic aquafici aquafici. I can never say that one right the first time. As well as a group called the chloroflexi and Deinococcus thermus. Fun fact: Deinococcus thermus. There's an organism in there. It's named Deinococcus radiodurans. It can actually survive enough as much radiation that's produced by an atomic bomb. It's kind of a crazy organism. But those are thermophilic, kind of tough to kill organisms. Then closely related to that are actually our gram-positive organisms, which include Bacillus and Clostridium. So Bacillus, um, Bacillus, for instance, we talked about anthrax earlier. Anthrax is Bacillus anthracis. It falls here. You might recognize Clostridium, such as Clostridium difficile, which causes C. diff, which is a really nasty bowel infection. So those are our low GC firmicutes. And then we also have what we call our high GC firmicutes, which I think are really, really cool organisms. They include organisms such as streptomyces. Now you might recognize strepto as in streptomycin, which is an antibiotic. But actinobacteria are really cool. They're found pretty heavily in the soil, and they're known for producing a wide range of antimicrobials, again, such as streptomycin, tetramycin, and so on and so forth. So really cool group of organisms. But again, these are our gram-positive bacteria. And this gram-positive idea will become important in just a minute when we talk about the test that helps us identify gram-positive bacteria. Now we have our spirochetes here. Now spirochetes are corkscrew-shaped bacteria. Now they're kind of a weird group of organisms, but one organism you might recognize from this group is called Treponema pallidum. Now Treponema pallidum causes syphilis, and it is a spirochete. So not a crazy diverse group of organisms, but again, medically important, as one of them causes one of the oldest diseases that we know, syphilis. So then we move into the photosynthetic organisms, and these include our cyanobacteria and a group of bacteria called chlorobi. Now, chlorobi is a pretty cool group of organisms. They're what we call green sulfur bacteria. You can Google what that means, but they do some cool stuff with photosynthesis in the absence of oxygen. So pretty cool group of organisms. And then we finally get into what you could argue is probably our most advanced group of organisms, which we call the proteobacteria. Now, of the 15,000 or so species of microbes, about 80% of all identified bacteria are proteobacteria. Now, within the proteobacteria, there's 5 main classes: beta, gamma, alpha, epsilon, and delta proteobacteria. Now, there's lots of interesting diseases and useful things that come from the group of proteobacteria. For instance, within the delta proteobacteria, there's a really cool bacteria called Delovibrio. It's known as a voracious hunter. It hunts other microbes. It's pretty cool. But lots of delta proteobacteria are known to be involved with sulfur cycling in natural environments. Epsilon proteobacteria, not a lot of them on the planet, but one important one is Helicobacter pylori, which is the causative agent of ulcers in humans. So kind of a cool group. We have alpha proteobacteria, which actually one of the cool things from the alpha proteobacteria is a group of bacteria called rickettsia. Now, rickettsia, you might recognize them as being a causative agent of rickets. So it's a skin infection called rickets, aptly named after rickettsia. But interestingly enough, this alpha proteobacteria, these rickettsia, are actually the most closely related ancestors to our own chloroplasts. So our chloroplasts, our own mitochondria. So we actually think our mitochondria come from the rickettsia. So kind of a cool thing. Now, gamma proteobacteria are also pretty cool. This is where E. coli lives and many, many other enteric pathogens such as Salmonella and Vibrio. They're gamma proteobacteria. And alpha proteobacteria are pretty cool. Now, beta proteobacteria, I jumped back to the alpha ones, but they're known to be some pretty important organisms for the health of soil and the health of plants as well. So those are our major groups of bacteria. We have our proteobacteria, our photosynthetic organisms, our spirochetes, our thermophiles, and our gram-positive bacteria. And then we have our two major clades of archaea. So that is not all the microbes out there. So I will post a I'll post a robust phylogenetic tree to our class's website so that you can see what actual robust tree of life of microorganisms looks like. But this is a pretty good basis of some of the major groups that we're going to be looking at and thinking about for this semester. Now, prokaryotes come in three distinct structures: bacillus, cocci, spirillum, which is another way of saying bacillus being rod-shaped, cocci being circular shape, and spirillum, which is where our spirochetes fall, as helical or corkscrew shaped. Now, these are the three major classes. There's not a lot of deviation from them, but there are some deviations from bacillus and cocci. You can have hooked bacillus or shortened bacillus or tetrad coccus, so like four coccus bound together. So there is some variability, but this is pretty much all the shapes you see for prokaryotic microorganisms. Now, prokaryotes have, as I mentioned, a cell wall. Now, cell walls are important, right? So they help maintain shape of bacteria. It helps maintain their internal environment to be distinct from their external environment. And so it's important for structural shape, but as with all membranes, a cell wall has to be permeable. And peptidoglycan, which is just a polysaccharide, so it's just a bunch of sugars, it basically looks like a chain link fence. And so it is very durable, both contractile as well as tensile strength, but it is permeable, so it allows things to flow through. Now, pseudomurin, which is only found in archaeas, it has a very similar structure, and again has that shape as well as the ability to allow things to pass through it. Now, within our organisms that have peptidoglycans, so our bacteria, we can actually use a technique called the Gram stain to identify the two distinct types of cell walls that we see in bacteria. Now, you might have noticed before when I mentioned the gram-positive and the gram-negative bacteria. So if you can go back to our slides here, our proteobacteria are going to be our gram-negative bacteria, and then our gram-positive bacteria are going to be this group here. And so we can use a test called the Gram stain to identify the differences in these two organisms. Now, gram-positive bacteria have a very thick layer of peptidoglycan and thus stain a purple color. And then the gram-negative bacteria have a very, very thin layer of peptidoglycan and thus stain a pink color. So let's dive into this really important microbiology technique. And you can see the results down here in the right corner, where we have a purple bacteria that's gram-positive and a pink bacteria that is gram-negative. Now, there are 4 steps to our Gram stain. There's a few in between, but these are the main 4 steps. So the first thing we do is actually apply a stain called crystal violet. And what this does is it stains both gram-negative and gram-positive purple, because crystal violet, as you can see, is purple. Then we apply what we call a mordant, M-O-R-D-A-N-T. Now, this mordant is called Gram's iodine. Now, the interesting thing, what Gram's iodine, it actually develops a crystal violet iodine complex inside the cells. And basically, it locks the color into the very thick peptidoglycan layer of gram-positive, but it doesn't do the same thing for gram-negatives. Then we apply alcohol in step 3. Now, what this does is it washes the Gram's iodine slash crystal violet out of the gram-negative bacteria. So remember, the gram-negative bacteria have that very, very thin cell wall. And it doesn't do anything to the it doesn't do anything to the gram-positive, because again, they have that very, very thick cell wall. And then we counterstain with a dye called safranin. And because the gram-negative bacteria have lost their dye because of the alcohol, they stain pink or reddish color. And the gram-positive, because they've just basically kept onto all this purple dye, simply don't change in color. And so we have this, again, this 4-step process where we can selectively identify those that have a thick cell wall, i.e. the gram-positive bacteria, and those that have a very, very thin cell wall, i.e. the gram-negative bacteria. Again, all based on the morphology of their cells. But let's look at what this looks like. So we have gram-positive bacteria on the left and gram-negative bacteria on the right. You'll notice that the gram-positive bacteria have that double layer plasma membrane, and then they have a very, very thick layer of peptidoglycan as their cell wall. They have these proteins called teichoic acids that anchor the cell wall to the cell membrane so the cell wall just doesn't float off. But again, that's what we have for a gram-positive, which is why they stain purple, they hold on to that dye really, really well. Then we have a gram-negative bacteria. Now you'll see they have that inner plasma membrane, a very, very thick layer of very thin layer of peptidoglycan, and then they have an outer layer, an outer plasma membrane as well. So they have a double lipid bilayer and a very, very small inner peptidoglycan layer. So pretty cool, cellular structure, again, can be detected with a very simple stain. Now, there are other things outside of having a membrane or a cell wall. Now, there are certain groups of organisms out there, mainly in our archaea, but we do see it in our bacteria that have what we call S layers. Now, these are very, very rigid, what we call paracrystalline layers. And basically, they're on the outside of the peptidoglycan or even the outer membrane layers in our bacteria. And basically, it's kind of like this sticky like substance that basically allows them to adhere to surfaces, allows them to attach to particles and things like that. Then we can also see in our bacteria what we call as a capsule. Now, these are these these like slimy gelatinous layers. You could picture it like if you've ever cooked like bacon or like any fatty meat in your house, and you leave the pan to sit, and that fatty layer forms on the pan. That's what a capsule looks like. It's gelatinous, gooey, fatty layer that aids them from attachment. But it actually, interestingly enough, in microscopic organisms that have it and that are pathogenic, it actually protects them from our own immune systems. So kind of an important thing for certain groups of pathogens. Now, as I mentioned, our bacteria and archaea can have flagellae. So flagellae are slender, rigid structures. They're helical. They're composed of a protein called flagellin, and they're involved with locomotion. They basically spin them like a boat's propeller, propelling a bacteria forward through a liquid environment. They can also have what we call pili. Now, pili are short hair-like structures that are found only in gram-negative bacteria that allow them to attach to other bacteria as well as to surface and aid in a process called conjugation, which we'll discuss in a little bit. So let's look at a flagella. Now we have a bacterial flagella. Now a bacterial flagella is powered by a force called proton motive force. So they basically it's like cellular respiration. They build up a proton gradient to spin this like a boat's propeller. But you'll see that in this gram-negative bacteria, it's anchored in not only the outer membrane, but the inner membrane and the cell wall as well. And again, it spins like a boat propeller, propelling a bacteria through an aquatic environment. And you might not think about like certain environments being aquatic, but bacteria always have to live in sort of an aquatic environment. Now, bacteria have evolved this really specialized coating, which we call an endospore. Now, these are actually really, really cool. Now, these very, very thick walls that form around the genome of a bacteria and some of their cytoplasm basically form in response to environmental stress. Now, this can be seen as heat or changes in pH or antibiotics. It can be any stress the bacteria sees, but the bacteria that produce these, which are our gram-positive bacteria, basically make these endospores, and they're actually one of the most resistant things on the entire planet. So you could basically take an endospore and put it into space, and you could bring that bacteria back from space, and you could germinate the endospore. So it's a really, really strong structure that allows these bacteria to survive in really, really stressful conditions. Now, there are some funky prokaryotic cells out there. Now, I mentioned they don't have membrane-bound organelles, but we can actually see some bacteria that have complex inner membrane systems. And so what they do is they take their cellular membranes and fold it in to form these inner structures, as you can see here. I don't think it's pretty easy to see here. You can see the rings of membranes here, but I think it's much better to see here on this right image, where you can see all these internal structures. And these aren't organelles or anything like that. These are just what we call invaginated regions of the cellular membrane. So basically they take their membrane and tuck it on the inside and tuck it inside. And basically what they allow these organisms to do is to have essentially a barrier, which makes it easier to perform respiration and or photosynthesis. So again, not an organelle, but some pretty interesting, complex internal structures for our microscopic organisms. Now, prokaryotes, as we mentioned, keep their DNA in the nucleoid region. So it's typically a single circular chromosomes. They can also have accessory chromosomes, which we call plasmids. And we'll talk about plasmids a little bit later on. But these chromosomes are anchored to the nucleoid region of the cells via a group of proteins. So that's our nucleoid region. Now we also have ribosomes, which are exactly the same as ours, similar structure, exact same process, but they are typically smaller. They have slightly different protein and RNA content, but again, functionally identical to what ours do, taking messenger RNA and making protein. And because they are distinct from our own ribosomes, many of our antibiotics we use actually selectively target ribosomes to basically kill off bacteria that are inside of our bodies. So that's all the structure. Now let's just dive into a little bit of the genetics of our prokaryotes. Now, they do not reproduce sexually, right? They reproduce by binary fission. And because of that, they lose out on all the benefits of sexual reproduction, right? From a genetic diversity perspective. So as such, bacteria and archaea have developed three modes by which they can get new DNA essentially from the environment or for another organism into their own genome. And these are called conjugation, transduction, and transformation. Now, conjugation is direct cell-to-cell contact, transduction is transmitted via viruses, and then transformation is DNA directly from the environment. And again, this is found in both bacteria and archaea. So let's talk about transformation. Now, first transformation was observed in the 1800s by Frederick Griffith. Now, he was playing around with an organism called Streptococcus. Now, you might recognize the first four letters, strep. If you ever had strep throat, you had Streptococcus growing in your throat. So it's kind of a jerk organism. I had strep throat when I was younger, about 60 times until they chopped my tonsils out. So I have a particular disdain for Streptococcus, though it was pretty sweet to stay home from school and watch The Price is Right all day because of strep. So I kind of have a love-hate relationship, I guess, is what I'd like to say about Streptococcus. So transformation is kind of a cool process. Now, when bacteria die, they release DNA into the environment. So their cells basically rupture and their genomes leak into the environment. Now, there are some bacteria that can undergo this process by which they pick up foreign DNA from the environment and they allow themselves to incorporate it into their own genome. This is what transformation is called, picking up DNA from the environment. So again, what does this look like from essentially from a mechanistic standpoint? Some bacteria dies, another bacteria that's still living says, hey, here's this piece of DNA, I'm going to uptake it, I'm going to incorporate it into my chromosome, thus now I have a piece of DNA from another cell. Now this is what it looks like just willy-nilly, naturally, but we can also do what we call artificial transformation, where we can physically force a bacteria to uptake a piece of DNA that's medically important to us. So for instance, for those of you that know anyone that's diabetic, this process of transformation is actually the way we make insulin in a industrial setting. We've basically forced E. coli to pick up a gene of interest, that i.e. making the gene for insulin. We forced the E. coli to incorporate it into its chromosome, and now we have E. coli making insulin for all the diabetics of the world. So again, a natural process, but also one we can do in a artificial setting for making cool things, such as insulin. Next up is transduction. Now, transduction is a pretty cool process, but this one is mediated by viruses. Now, transduction can basically transfer any gene, and it occurs during accidents during what we call the lytic cycle. So when we talk about viruses later on this lecture, we'll talk about what the lytic cycle is, but just know transduction happens during the lytic cycle. And what ends up happening is, as a virus is making more copies of itself, it accidentally inserts bacterial DNA into its chromosome and then transfers it to a new bacteria in a subsequent infection. So viruses horizontally transferring DNA from one bacteria to the next. Now, that's generalized transduction. Now, we also have what we call specialized transduction, which is where which happens due to accidents that happen during the lysogenic cycle. And again, we'll talk about these two processes later on in the lecture. But basically, this is the imprecise excision of what we call prophase DNA. And this is just, when we say phage, it's a virus that infects bacteria. Pro, meaning it hasn't left the genome yet. And again, we'll get into what this means later in the lecture. But these phages can carry both phage genes as well as chromosomal genes from their bacterial host. So let's look at this. So we have an infection with the phage. The phage lands on the bacteria, injects its DNA into the cell. The phage DNA is replicated, and the host DNA is then degraded. And then the phage particles are packaged with DNA and then are released. Now, you'll notice that the phage itself has some bacterial DNA and some viral DNA as well. Then they go on to infect a new host. They inject their DNA again, and then the DNA is then incorporated into a new host, and the cell thus now contains DNA from a donor, again, all mediated by virus. So you see the original chromosome was blue, and now we have that blue chromosome down here in the the eighth panel here. So that's how transduction works. It's a pretty cool process, medically very important. It doesn't just happen to bacteria. I'll just make mention. As I mentioned in our genome evolution lecture, human DNA has about 7% virus, and that 7% virus comes from transduction. So not just a bacterial thing, but also a eukaryotic thing as well. Now, the final way bacteria can acquire novel genetic material is via conjugation, which you could basically classify as bacterial sex. And this is cell-to-cell contact where they transmit a small circular piece of DNA, which we call a plasmid. Now, this has been best studied in E. coli, so gram-negative bacteria. Now, in E. coli, conjugation is based on the presence of what we call an F plasmid, or what we call a fertility factor. Now, those cells that are F positive contain plasmid. Those that do not contain a plasmid are F minus. So the way this works is that the F positive cell, i.e. it has the plasmid here, produces an F pilus, which you can see in this picture here, but basically think of a F pilus as a bridge from one bacteria to the next. And this basically connects it to the F minus cell. Then we have the transfer of the F plasmid, so this piece, into through this pilus, which is now called a conjugation bridge. Now, this F plasmid is copied by a process called circle replication. We're not going to talk about what circle replication is, but that's how you copy plasmids. And ultimately, it results in two bacteria, one that was originally F positive, one that was originally F negative. Now both are F positive because they both have that fertility factor on that plasmid. And this might not seem like kind of a an important thing, but it is relatively common in the medical world. Many bacteria that are antibiotic resistant have required their antibiotic resistance genes via conjugation as a process. So again, this direct cell-to-cell contact for our bacteria, really important way to transfer genetic information. Now, the F plasmid can also integrate into the bacteria's chromosome, which is actually kind of an interesting thing because it's very similar to the way crossing over works in eukaryotes. So if you remember from meiosis 1, crossing over is a type of recombination that we see in eukaryotes. So it's a similar process, which is pretty cool. Now, we can also have a special group of organisms, which are called HFR cells, which is high frequency of recombination cells, where the F plasmid is directly integrated into the chromosome and thus replicated every time the host divides. So the plasmid gets passed on from generation to generation to generation. And interestingly enough, these HFR cells, the F plasmid can also excise itself by reversing the integration and then basically basically go on to allow the bacteria to conjugate again. So kind of a fun thing. And this is what this looks like. We have an F positive cell. There's sort of standard thing, right? An F positive cell becomes an HFR cell via this plasmid linking up and basically integrating itself into the bacteria's chromosome. And then an HFR cell, can excise its own plasmid. Again, it's kind of like tying a knot almost, as you can see in this image, thus releasing the plasmid, allowing the bacteria to then go on and have another round of conjugation with something else. Now, during conjugation in our HFR strains, the transfer of genes is actually linear and progressive. And what do I mean by that? Well, genes farther from the origin of transfer, i.e. this red part here, will be transferred later on. And then there are different genetic markers that appear in the recipient cell at specific times. So basically we can use this idea of homology between the plasmid and the chromosome to map based on how long it takes for them to enter into the host chromosome. So we can map our plasmid to know exactly what's on our plasmid. So fun old technique to basically understand how plasmids work. Now, one common aspect, and I've mentioned this a few times in this lecture thus far, is antibiotic resistance. And these idea of antibiotic resistance, or being not susceptible to an antibiotic used by humans, is commonly carried on, on plasmids, which we call R or resistance plasmid. And basically, the way R plasmids work is they're transferred the same way an F plasmid is. But basically, an R plasmid acquires resistance genes via a process called transposable elements. So we talked about that in the genome evolution lecture. Now, these R plasmids are really important in the emergence of resistant strains of Staphylococcus aureus. So you guys might have heard of MRSA, or methicillin-resistant Staphylococcus aureus. And it's a really nasty bacteria that pops up in hospital environments. But this organism, again, which causes hundreds of thousands of infections every year, got its resistance from an R, or resistance plasmid. Now, plasmids are a great way, but we can also see transduction as a way to, i.e. viruses, as a way to transfer pathogenic genes or resistance genes. Now, there's a really cool example here in E. coli. Now, you guys might have heard of E. coli 0157H7, which is known as the Jack-in-the-Box E. coli. So back in the 90s and 2000s, there was a strain of E. coli that popped up in the Jack-in-the-Box restaurants that killed a couple dozen people. Well, how did that pathogenic E. coli come to be? Well, it actually got its pathogenic genes from a virus. So it converted it from regular happy E. coli into this nasty strain via viral transduction. So lots of cool ways these bacteria can get these genes. Not always the best for us as individuals. Now, as with eukaryotes, mutations can arise spontaneously, as with everyone, right? And we could think of chemicals or radiation that increase the likelihood. So mutations are pretty common, and as we discussed, mutations are partially the way new traits pop up. Now, we have a group of bacteria which we call auxotrophs. These are actually what we call nutritional mutants. They actually cannot survive on what we call minimal media because of mutations. So minimal media is like basically the bare bones. It's like sugar and nitrogen and phosphorus, and that's pretty much it. And these mutants have popped up spontaneously that makes them actually unable to survive in these really, really simple environments, which they actually naturally should be able to. Now, mutations, as well as plasmids, which can carry mutations, actually can spread pretty rapidly in the population. Now, we see this with methicillin-resistant Staphylococcus aureus, or MRSA. We're also seeing it in another Staphylococcus aureus, which we call VRSA, or vancomycin-resistant Staphylococcus aureus. But mutations, once they happen in bacteria, if they are beneficial, can actually spread really rapidly throughout these populations. Now, very much like us, bacteria have an immune system. And I know that's kind of a weird thing to think about, right? We think of our immune systems being large and complex, but bacteria have their own immune system. Now, this immune system is what we call CRISPR-Cas9. Now, it's an immune system not for fighting off other bacteria, but it's an immune system for fighting off viruses. Now, CRISPR is pretty cool. And basically, when you look at a bacteria's genome, you can see these repeated sequences with spatial regions, which are called CRISPR, or clustered regularly interspaced short palindromic repeats. I never remember what CRISPR stands for, which is why I always have to write it down when I talk about CRISPR. But basically, it's kind of like almost like a B cell for bacteria. So you guys might know from immunology, if you've ever taken it, that a B cell is kind of your memory cells. And so once you experience an infection, you develop B cells, which allows you to respond to their infection when you're exposed to it again. And basically, CRISPR, which are just segments of the genome, are like B cells. So if a bacteria gets infected with a virus and survives, it basically incorporates a small piece of that virus's genome into its chromosome, into these CRISPR regions, and allows them to say, hey, here's this virus again. My CRISPR regions tell me I've been exposed to this virus. Thus, I can basically recognize that virus and then degrade that viral nucleic acid to allow the virus not to infect me. So pretty cool immune system, very similar to the way our immune system works. You have a memory, it recognizes the pathogen and kills the pathogen before it can cause a problem. Now, CRISPR, as you might have heard, is also useful for gene editing. Now, you guys might have heard in 2019 of the CRISPR babies that were born in China, where they edited the genome of I think it was just two infants or two, babies that weren't born yet, to make them slightly to make them immune to HIV, if I'm not mistaken. So really, really powerful genetic technique to directly edit genes of interest in a wide variety of organisms. So that's structure, that's genome. Now let's talk a little bit about the metabolism of prokaryotes. Now we saw that the shapes of prokaryotes are very simple, right? They're cocci, rods, or spiral shaped. But where prokaryotes really shine in terms of their diversity, outside of species level diversity, is actually their metabolism. Now, we can group bacteria, as with all organisms, into two broad categories of how they get their carbon. So the first is autotrophic organisms, and now these are organisms that derive their carbon, i.e. their food, from inorganic CO2. Now, unlike plants, which get their energy from the sun, which are called photoautotrophs, there's a group of bacteria which we call chemolithoautotrophs that actually derive energy from oxidizing inorganic substances such as hydrogen and sulfur, as well as some nitrogen as well. And so instead of sunlight to power photosynthesis, they use chemicals to power photosynthesis, which is why they're chemoautotrophs. Chemicals to power their autotrophy, as opposed to photoautotrophs, which use the sun to power their autotrophy. So pretty cool group of organisms. Then we have our heterotrophs, which is basically where we fall, where we get our we get our energy, our food, from organic molecules produced by autotrophic organisms. Now, there are two groups of heterotrophs that are kind of unique to bacteria. Now, we are just regular heterotrophs. We eat food, we break it down, we respire. We're just regular heterotrophs. But there are two special groups of heterotrophs in the bacteria world, which are called photoheterotrophs, which actually capture light as energy, and they use that energy from the light to break down organic carbon made by organisms such as phototrophs or chemotrophs. So pretty cool. And there's also what we call chemoheterotrophs, where they get both their carbon atoms and energy from organic molecules, and that's where we fall. And there's a to use chemicals to get their energy from. So like sulfur and nitrogen and things like that. So lithoheterotrophs. That's our third class. Pretty cool. Now, we also have organisms that rely on other types of metabolism. So let's talk about those a little bit. Now, prokaryotes can do a wide variety of respiration and fermentation processes. Now, as I mentioned in the previous slide, eukaryotes are restricted to organic electron donors and oxygen as their terminal electron acceptor. So that's all we can do. But interestingly enough, prokaryotic fermentation can actually metabolize pyruvate as well as other compounds to recycle electron donors. Now, you might recognize pyruvate as being the end product of glycolysis, which you would be absolutely right about. And so prokaryotes use that. Now, interestingly enough, fermentation, the byproducts of it are actually industrial and commercially viable. Now, for instance, for those of you that like yogurt, for those of you that like kombucha, for those of you that like sauerkraut or kimchi, or any fermented food or drink on this planet, this prokaryotic fermentation is actually a really important part about producing many of the tastes and flavors that are important in fermented foods as well as fermented beverages. So just as a note, if you're like me and you like kimchi a lot, you owe it all to this kind of odd fermentation pathway we see in our prokaryotes. Now, our prokaryotes are typically heralded as being bad for us, right? I kind of preface this class by saying, humans and microscopic organisms have gone hand in hand with one another for a very, very long time. And part of that is they're pathogens. But the reality is most prokaryotes on this planet are beneficial to us either directly or indirectly. Now, one of the common things we see in the environment is decomposition. Now, bacteria, as well as fungi, which we'll talk about in next week's class, are really, really important for the decomposition of dead organisms, releasing the energy back into the environment and the nutrients back into the environment. So, a really important part of what we'll talk about later in the semester, which is called the carbon cycle. We also see microscopic organisms are really important as photosynthesizers. So every breath you take on this planet, about 50% is derived from plants, so trees and bushes and all that stuff. And the other 50% of the oxygen that you breathe every day is derived from prokaryotes. So they are really and really important for the generation of oxygen on this planet. And as we talked about as with the development of the Earth, ancient cyanobacteria, which are just bacteria or prokaryotes, added oxygen to the planet, thus allowing oxygen to start forming on our planet. So photosynthetic prokaryotes, super duper important for the evolution of Earth, as well as the average everyday person's life. Now, there's also a group of bacteria, which we call nitrogen fixers. Now, they take all the nitrogen in the air, which is N2, and they do a process called nitrogen fixation, where they fix that N2 to ammonia, or essentially fertilizer. And you might think, okay, well, why does that matter? Well, as we'll talk about later in this semester, this is actually the only way nitrogen can get into the environment. This process of nitrogen fixation, which is solely catalyzed by groups of organisms such as rhizobia and anabina, because without these organisms, there would be no nitrogen. And you might say, okay, why do I care if there's nitrogen? Well, remember, your DNA and your protein are about 50% or so, I'm sorry, about 25% or so nitrogen. So without these microscopic nitrogen fixers, there would be no nitrogen, thus no DNA or protein, thus no life. So really, really important. Now, in addition to microbes living on and inside you, plants actually have developed and have evolved a really long-standing relationship with many, many microscopic organisms. Now, there's a really cool group of organisms that live in inside plant roots, which are called nodule forming or rhizobia, and they live on plant roots and they fix nitrogen for the plants. But there's a wide range of plant associated bacteria that help plants grow better. Now, we owe so much of our agricultural productivity as a species to the natural bacteria that live in our soil and thus help plants grow better. In addition, we also know there are many parasitic relationships between bacteria and their plant counterparts, including, pathogenic disease such as rots or such as, example I hear is crown gall disease. So if you ever go to, in the woods or in a park and you see a big, like kind of like looks like a tumor growing on a tree's, trunk, that's crown gall disease. Again, caused by a bacterial pathogen. Now, as I've also mentioned, we are hosts. We are our own microbial ecosystems, as are dogs, as are cats, as is everything that lives on this planet. There are mutually beneficial associations between us and our bacteria. And they do a wide variety of things, but I'm just going to focus on what they do for humans, simply because they're really important for what they do. But for us, they help protect us from pathogens. So if you had no bacteria on your skin, you would get skin infections constantly. And if you had no bacteria in your gut, you would get gut infections constantly. They also help train your immune system. So they make you less essentially reactant to things. So if we didn't have bacteria, we'd have allergies for days. They also produce many of our vitamins. So vitamin B and vitamin K, all the vitamin Bs, of course, not just any one B, but all the vitamin Bs and vitamin K we basically rely solely on our bacteria to give us in addition to our diet. And we also see our microbes are important for a wide variety of things, such as helping us digest things, helping us, helping us maintain healthy skin. We owe our own scent. So when you sweat, your scent of your sweat, you actually owe to your organisms, your microscopic organisms, as they break down the products of your sweat. So that's just a short little mention of some of the things microbes do for you. But just know, without your microbes, you could not live. Without your microbes, your cat couldn't live, or your dog, or your hamster, or whatever it is. Microbes and animals are essentially like plants tied together inextricably. Now, bacteria are also useful in a process called bioremediation. So basically what we're saying is removing pollutants from water, air, and soil, which is kind of cool. Now we can look at two distinct types of this, which we call biostimulation, where we add nutrients to encourage the growth of naturally occurring microbes. So we've seen this after every oil spill. After every oil spill, if we look in the sediments near where oil spills occurred or underneath an oil spill, the bacteria that can break down oil essentially expand in their activity and their abundance, thus allowing us to better remove oil from the environment, but clearly not a substitute for just not spilling millions of gallons of oil into the environment. Then we can also have what we call halo respiration, where the bacteria remove halogenated compounds from toxic waste, so making toxic waste slightly less toxic, but still toxic. So lots of cool things. Now, as I mentioned, outside of being nice to us, bacteria have been a great check on our human population since day one. Now, in the early 20th century, so talking about the 1900s, because that was so long ago, 21 years ago now. But in the early 20th century, infectious disease actually killed about 20% of children before the age of 5. So that's a really high mortality rate. However, sanitation and antibiotics have considerably improved the situation. How interestingly enough, though, we've seen a pop-up of many bacterial diseases reappearing in our society. So, for instance, measles is popping back up, mumps is popping back up. We're starting to see diseases that we almost eradicated from this planet starting to pop back up as they become immune or people start to be lax on their sanitation or vaccine use and so on and so forth. But I figured I'd give you this handy dandy table where we have some common diseases, well, some cool diseases, I should say. They're not super common, but some diseases that are pretty cool, from anthrax to diphtheria to leprosy, what the pathogen is, what their vector or reservoir is, and what the epidemiology is. Take a peek at this. If you have a few minutes, it's a pretty cool little table. And this table continues. We have some more Lyme disease, plague, pneumonia, and so on and so forth. But let's talk about how bacteria cause disease. Well, to cause a disease, a pathogenic bacteria needs a couple things. It needs to enter your body. It has to colonize the site of infection. It has to evade your immune system. It has to spread to other sites in your body. And it has to cause damage by the production of toxins or by triggering inflammatory processes or your immune system kind of going crazy. And so to do this, bacteria have structures or physiological processes so that they can use the host as environmental niches. Now let's talk about an example of this. We're going to look at tuberculosis. It has been a scourge on the population for thousands of years. It still kills thousands of people every year. So it's still around today, but it's been doing its thing to humans basically since day one. Tuberculosis is a really old disease. Now, this disease is caused by Mycobacterium tuberculosis. Mycobacterium are pretty cool. Now you might, if you know your Latin, you'll recognize myco is essentially like fungus, right? So these are fungus-like bacteria. So they're pretty cool organisms. Now TB affects your respiratory system and is easily transferred from person to person through the air. Um, Now tuberculosis we got under control in the 70s or so with an antibiotic called rifampin, as well as a few other different antibiotics. But we're actually starting to see a reemergence of what we call multi-drug resistance or MDR strains of Mycobacterium tuberculosis, which if you think about if it's been a scourge for thousands of years and we pushed it back and now it's mutated to be dangerous again, well, it's going to continue to be a scourge going forward. Now, Helicobacter pylori is another great example of a pathogen. It's an epsilon proteobacteria, so it's one of our gram-negative bacteria. Now, if you haven't heard the story of Helicobacter pylori and how it was discovered, I suggest you look it up. It's a pretty cool thing of a crazy scientist who drank Helicobacter pylori and took antibiotics to prove that Helicobacter pylori was the causative agent of stomach ulcers. Really cool disease. I recommend you take a peek at the history of it. But Helicobacter pylori is a bacteria that lives in your stomach. It causes stomach ulcers. Part of the way it does is it increases the amount of acid production, and you can treat stomach ulcers by actually reducing stomach acid with medication. Now, Helicobacter pylori can also cause gastritis, so essentially inflammation of your gastrointestinal system, which you can treat with antibiotics. It's also been associated with stomach cancer and certain types of lymphoma as well. But again, this bacteria has to colonize, evade your immune system, and cause harm to your body some way for it to be pathogenic. Now, since we're all in, in a college environment, I'd be remiss if I didn't discuss STDs or STIs, which are sexually transmitted diseases or sexually transmitted infection. Now, gonorrhea and chlamydia are two great examples of this. We talked about AIDS in our genoevolution class, but we'll discuss two other sexually transmitted disease. Now, gonorrhea is a interesting organism. It's caused by the organism Neisseria gonorrhea, which is aptly named, and it's actually one of the most prevalent and communicable diseases in North America. Now, it is exchanged through exchange of bodily fluids, so blood and semen, as well as vaginal fluids, and those have to hop into open wounds. That's kind of how STIs work. It can also pass from the mom to the baby via the birth canal. Now, gonorrhea is an interesting disease. It's not particularly very dangerous. It's gross, but it's not going to likely to kill you. However, there is actually new strains of gonorrhea popping up, which doctors have termed super gonorrhea, which are resistant to multiple types of antibiotics and are actually becoming much, much more dangerous to human health. Then we have our next organism, which is chlamydia, which is caused by chlamydia trachomatis, if I believe that's caused right. Now, for the longest time, chlamydia was called the silent STD because it's not a super dangerous one. It doesn't have the craziest of symptoms, but interestingly enough, its incidence has skyrocketed lately, as has gonorrhea, which is kind of an unfortunate thing, but it's happening. But chlamydia can cause, outside of its normal symptoms, pelvic and inflammatory diseases, as well as heart disease. So involved in multiple systems, not just your sexual areas of your body. And we can actually look at this over time. And I think this is a pretty easy to understand graph, but we have years on the x-axis, so 84 to 2014, and the number of cases per 100,000 people. You'll see that gonorrhea has gone down a bit, but it is starting to peak up again. Actually, if you look at this graph nowadays, you'll notice that gonorrhea is coming back up again, which is kind of unfortunate, but it is the way of the world. And you'll notice that chlamydia is really skyrocketing, though chlamydia has come down a little bit in recent years. It's not as increasing as dramatically as it was before. And then we can see that our syphilis, again, is kind of the steady background thing. Syphilis is a cool disease. Syphilis can travel from your genitalia up to your brain in late stage infection and hang around in your brain for decades, slowly eating away at your brain tissue. So again, these organisms, microscopic, causing sexually transmitted diseases, have varying levels of dangerousness, but again, can affect multi-systems inside your body. So that's our prokaryotes. Let's switch gears. Let's talk about a topic that is near and dear to your heart right now, and that is viruses. And it's near and dear to your heart, not because you want it to be, just simply because it has dramatically affected your life. And, that's all I have to say about that. I'm sure, like you guys, after 1 year of the pandemic, I'm tired of it. You're probably tired of it, but it's going to be our future of our life going forward. So let's talk about those little teeny tiny buggers that cause COVID and other diseases like measles or HIV. Let's talk about our viruses. But viruses are simple, which kind of is a good and a bad thing at the same time. They have nucleic acid, they can have DNA or RNA as their genome, and this nucleic acid core is surrounded by protein. There's no cytoplasm, and viruses aren't even a cell. So they don't have a membrane, they don't have cytoplasm, they don't even have ribosomes. They are just protein surrounding DNA or RNA, and that's all they are. So there's this kind of back and forth narrative we see among scientists, and it's been going on for probably about 2 decades now, about whether viruses are living. And they don't fit all the traditional, definitions of life. But at the end of the day, viruses exist on the continuum of maybe they're living or maybe they're just protein floating around infecting things. We don't quite know yet. And we don't know their evolutionary origins quite yet as well. So, but let's talk about their structure. And then let's talk about how they live their lives. So, as I mentioned, they're DNA or RNA-based organisms. They can have circular chromosomes or linear chromosomes. Their DNA or RNA can be single or double-stranded. And RNA viruses can have segmented chromosomes, so they can have multiple pieces and not just one circular or linear strand of nucleic acid. So RNA viruses do some additional layers of things. And we classify our viruses based upon what we call the Baltimore classifications. And one of the biggest things in the Baltimore classifications, which I don't expect you to ever look up or know what it means, but we classify them by based upon their genome, which is DNA, RNA, or retroviruses. And we'll talk about all those soon. Now, as I mentioned, all viruses, or nearly all viruses, I should say, form a protein sheath, or what we call a capsid, around their nucleic acid core. Now, all their capsid is basically one or two proteins, sometimes three or four, but generally one or two, that are just repeating over and over and over again, forming an outer layer, essentially like a pseudo membrane. Not quite a membrane because it's not permeable, but forms an outer layer, which is you know, pretty pretty straightforward and simple, but clearly very effective. Now, some viruses also store specialized enzymes with nucleic acid cores, such as reverse transcriptases, and we'll talk about this. We're going to talk about HIV later. HIV has reverse transcriptase, which is a specialized enzyme with it has nucleic acid as its core. So, just as a note. And many animal viruses have an envelope. Now, COVID-19 is an enveloped virus. Now, an envelope is basically a piece of the host's cellular membrane that has viral proteins embedded inside it. So let's look at a few structures of viruses. So we have that capsid. Now, this is the tobacco mosaic virus, or TMV. It infects, as its name implies, tobacco plants. It's a really long, heavily studied plant because tobacco is a pretty, viable cash crop out there. But you can see it's got a capsid, again, repeating elements of protein over and over and over again. Its shape of its capsid is helical, so it kind of looks like a can of soda or like a glass or something, or a can or something like that. And then it has an RNA genome that's surrounded by this capsid. So pretty straightforward. We can look at the animal virus, which is our adenoviruses. Now, a little bit more complex. It's got what we call an octahedral shape. So it's got like this. It's almost like one of those crazy 12-sided dies. But again, repeating units of protein forming the capsid are surrounding the inner DNA core. Clearly, this little piece isn't missing in normal viruses. This is just to show you what it looks like. But you also notice it has these kind of funky spike proteins hanging off as well. That allow it to interact with its host. So that's our octahedral viruses. Now we have our bacterial viruses, or our bacteriophages, which I think look like lunar landers. You know, if you guys ever seen the Apollo lunar landers from the 60s and 70s, I think it looks like this. But you'll notice it has this tail, it has this base, and has these kind of funky spider-looking legs, and then it has its capsid up here, and then it houses its DNA here. So the head is octahedral, and it has a helical tail with these, again, kind of these funky spider-looking legs. As a note, these just help for attachment. That's what the legs do here. That's the little thingies. I'm forgetting their name off the top of my head now. These little things here help the virus do there. So let's look at another human virus. We have influenza. Now you'll see the capsid here in the center, surrounded the RNA, but you'll notice we have the envelope. Now inside the envelope, we have a couple what we call antigens. Now these are viral proteins. And remember, this envelope is basically your own membrane. So if you are infected with influenza, you're actually producing membranes, viruses with membranes that are your own membranes with viral proteins embedded inside. So that's what influenza looks like. So that's the gist of sort of viral structures. There's lots and lots of them out there. You can Google viral structures and you'll see ones for COVID-19, you'll see ones for Ebola, the cold virus, and so on and so forth. There's lots of cool shapes viruses take. But that's the basic structure. Now viruses, they're parasites. That's what they are at the end of the day. They rely on a host for energy, and they rely on a host for reproduction. And so our viruses are obligate intercellular parasites. Let's break that down. Obligate means they have to do it. Intracellular means they're inside of your cells, basically. And parasites, meaning parasites, right? They rely on you. So viruses are obligate intercellular parasites, meaning they rely on a host for all their needs. They can't live without a host. Now, viruses are really wild. Now, we know that there are 10 to the 30 bacteria on this planet. There seems to be about 10 to the 31 viruses, which is an astronomical number, but my point of telling you that was with with every host, there seems to be thousands, if not tens of thousands of viruses associated with every single living species on this planet, from the biggest organisms down to the smallest. Viruses are super diverse like bacteria. And there's no known species on the planet Earth today that doesn't have a viral pathogen associated with it. So just as a note, everything has viruses. Now, viruses, typically speaking, have what we call a host range, which are the types of organisms that they can affect. Now, as a general rule, most viruses has a very, very limited host range. So, for instance, the viruses that infect us only infect us. So, uh, the strains of influenza, the strains of COVID-19, the strains of the cold virus that infect us generally only infect us and nothing else. That being said, there are some viruses that have a wide host range, so they can infect humans and pigs and chickens and so on and so forth. But the vast majority of viruses, they go, I like this host. This is the only host I can really interact with. And part of that is the way viruses infect a host. They recognize the host cell types, and they recognize the host cell types by the proteins found in the host membrane, which are different between hosts. So if a virus can't recognize a protein inside your membrane, it can't infect you. So just as an now, there's also some viruses that exhibit what we call tissue tropism. Now, once they are inside of hosts, the virus can actually only infect certain tissues. Now, COVID-19 doesn't exhibit tissue tropism. It seems to affect your brain and your kidneys and your heart and your lungs. But you can think of something like influenza, which can only infect your lungs and nothing else. So, two distinct things. Because again, COVID-19 is not the flu, right? It's very, very distinct from the flu. Now, viruses can maintain what we call dormancy for a very extended period of times. So we can see this with chickenpox. So if you get infected with chickenpox when you're a kid, you might, as an adult, you might actually see chickenpox pop up again, but now it's not chickenpox, it's called shingles. So kind of fun. Now, how do viruses reproduce? So let's get into that. Now, viruses have to infect a host, and basically all a virus's genome tells you, or the organism or the virus, whichever person is analyzing the genome, I suppose, um, is basically just a set of instructions for replication. Now, what a virus genome does is it tricks the host cell into making new viruses. So basically, it comes in, hijacks your cells to make new viruses, which is pretty cool, right? It's like you buy a house and then you turn it into a factory. That's what viruses are basically doing. Now, cells that have viral infections are often damaged by the viral infections, and viruses have to reproduce only inside cells, not cells, because once they are outside of a cell, they're basically just protein hanging around. That's basically all viruses are. They're metabolically inactive, and they actually have a specialized term, which we call virons. So when they're outside of a host, they're virons. When they're inside of a host cells, they're viruses, just as a note. So viruses lack their own ribosomes, they lack their own enzymes and other uh, proteins that they need for producing more virus. So the virus hijacks the cell's transcriptional and translational machineries to express three classes of genes: early genes, middle genes, and late genes. And we'll get into those in a second. But ultimately, they come together to assemble and release the virus from the host. Now, viral shapes are kind of diverse, but there are two basic simple shapes that we see: helical and iso, ico, isosahedral, or generally spherical. But the morphology within these capsids is pretty diverse, as we've mentioned. So we can have naked viruses, complex viruses such as pox viruses, or even bacteriophages. And there are other types of envelope viruses. But again, viruses take some pretty interesting shapes. And you can see, again, pretty cool shapes. Complex virus with some symmetry around it. But viruses, they have different shapes, but they're generally pretty small. So we have streptococcus, which is a pretty small bacteria. Then we have a eukaryotic yeast cell. And you'll notice that viruses are, and we have our E. coli here. What you notice is that viruses are pretty small, right? 250 nanometers all the way down to 15 nanometers for a protein, right? So viruses are almost as small as some proteins. So the West Nile virus, for instance, 22 nanometers, about the size of the human hemoglobin protein. So very, very small, lots of diversity. Now, that being said, there are viruses out there which are called viruses, M-I-M-I, then viruses, of course, which are about the size of streptococcus here and larger, of course. So both viruses are incredibly small. Some are very, very large, but again, most of them are teeny tiny, some as small as proteins. Now, viral genomes differ greatly in both the nucleic acid as well as the number of strands. Now, most RNA viruses are single-stranded. So these include influenza, measles, the common cold, and they are replicated inside the host's own cytoplasm. Now, RNA viruses, very much like COVID-19, their replication is very, very error prone, i.e., there are high rates of mutation. And this actually makes it a bit difficult. When you have high rates of mutation, it makes it difficult for drugs as well as the host immune system, as well as vaccines to target those organisms and kill them. Again, all due to high, um, all, all, all high levels of mutation makes it a bit difficult. Now we have retroviruses, which HIV falls into this category, which is viruses that are RNA viruses. They're single-stranded RNA genomes that's then transcribed from RNA to DNA. So it's a process called reverse transcription, because regular transcription is DNA to RNA. Reverse transcription is RNA to DNA. And so the enzymes that carry this out are enzymes called reverse transcriptase. And basically, this is our retroviruses. HIV is a type. There are other types, but HIV is the most clinically important for retroviruses. DNA viruses, as I mentioned, can be double or single-stranded, but their DNA is replicated in the nucleus of the eukaryotic host cell, I should say. And these include organisms such as smallpox, which we've eradicated from the planet, but it is a DNA virus, and herpes simplex. So those of you that have ever seen someone with a cold sore or have had a cold sore, you get a DNA virus called herpes. Fun stuff. And so here's some just a handy dandy table, couple tables, I should say, the same table but slightly longer, of a couple different things. Chickenpox, hepatitis B, herpes, AIDS, so on and so forth, and just some viruses, what they look like, what their genome is, and their vector, as well as epidemiology. Ebola is my favorite. Really, really scary disease. With peak mortality between 50 to 90%. But really, really cool looking group of filoviruses or filoviruses out there. Now, as I mentioned, there are giant viruses out there, and these are what we typically call our viruses, but there's also Pandora viruses, pittaviruses, and closoneuraviruses. I believe I said that right. And these are giant viruses because they essentially challenge our assumption about viruses due to their size as well as their genomes. Now, their size is significantly larger. Now, the average virus range from 20 to 250 nanometers, as we've seen. However, the virus can actually have a viron 750 nanometers, sometimes all the way up to 1.5 micrometers in diameter. So it can be the size of a bacteria. Its genome is 1.2 megabases or 1.2 million bases, and that might seem like a big genome, but that's not a big genome at all. You might recall from our genome lecture, the human genome has 3 billion bases, and the average bacteria has about a 2 billion base pair, 2 million base pair genome. So the virus, same size as a bacteria, about the same size genome as a bacteria. It was originally identified as a bacteria, but we actually know the virus and the other viruses down here are now considered viruses. And it's kind of a cool thing. Their genome actually shows the presence of translation machinery parts. So we actually think these giant viruses used to be bacteria, but they've essentially de-evolved to be viruses. So as I mentioned, we classify viruses based on a couple key areas. Now, the first is which we call taxonomy. Now we can use ICTV, or the International Committee on the Taxonomy of Viruses, which use the same sort of classification hierarchy that we saw in our first or second lecture this semester, where we use order, family, subfamily, and genus, and potentially species to classify a bacteria. We also classify the bacteria based upon the disease they cause, the host they're able to infect, as well as their genome expression, which is double-stranded DNA, single-stranded DNA, which I have referred to before as the Baltimore classification. So this is how we classify our viruses, because we have to classify everything. Now, classifying a virus based upon its disease or its host is pretty limited, because not all viruses cause diseases. Right? You don't have to have a virus causing a disease for it to be a virus. Right? It just doesn't have to work that way. Some viruses can cause different diseases under different conditions or during different times during infection. We saw that with chickenpox, right? Chickenpox as a kid, shingles as an adult, two very different diseases. So viruses can have a decent amount of variability in them. Now, some viruses, such as the common cold, can actually be caused by several different types of viruses. So we have one disease caused by multiple strains or multiple different types of these viruses. And there are some viruses that can infect very many different types of organisms. So viruses that are related to influenza, as well as, like SARS viruses, so the severe acute respiratory syndrome viruses, can infect many different types of organisms. Now, classification by genome expression is actually a little bit better. It's more broadly applicable than other classification types. Now, this classification by genome is, again, the Baltimore classification, and this sorts viruses based upon the relationship between their genome structure and their genome expression. And as we see advances in genomic technology, in particular what we call metagenomics, we've actually revealed really, really important roles for viruses in ecology and evolution of biological systems, both naturally as well as in hospital and environments, hospital and human-associated environments. Now we can look at the Baltimore classification. We have group 1 through group 7. Now group 1 is DNA, group 2 is just is DNA as well. Group 3 is RNA. Group 4 is RNA. 5, 6 are both RNA, and group 7 is DNA. And they all do slightly different things. Now, group 1 directly transcribes its DNA to messenger RNA. Group 2 converts its single-stranded DNA into double-stranded DNA and then transcribes it into RNA. Group 3 has to essentially change their messenger RNA into a single-stranded RNA, but it again directly makes messenger RNA from it. Group 4 converts its positive strand of RNA to negative strand of RNA, then to messenger RNA. Group 5 just takes its messenger RNA and uses it directly. So it's kind of a fun organism. Group 6 is our retroviruses. So they take their plus-stranded RNA, reverse transcribe it to DNA, then use that DNA to make messenger RNA. And then finally, we have group 7, which takes their DNA, converts it to plus-sense RNA, uses reverse transcription to make DNA, and then use that DNA to make messenger RNA. Group 7 is a dumb group of organisms, in my opinion, but that's how we classify organisms based upon their genome using the Baltimore classification. Whew. Now, bacteriophages, as we discussed, are viruses that only infect bacteria, and they're pretty cool. They're the ones that look like the lunar lander. They have those, they look weird and funky. But they are diverse, and they're only united by their bacterial hosts. Now, we call bacteriophages short just phages. And the best studied viruses are those that infect E. coli, and these are actually named the T series of bacteria, T1, T2, so on and so forth. But there are no microorganism on the planet that doesn't infect there are no bacteria on the planet that do not have a bacterial pathogen. So it doesn't matter if you're a virus or a bacteria, every bacteria is subject to predation by a virus. So just as a note. So that's classification, that's structure. Let's talk a little bit now about the life cycle. Now, the first one we're going to talk about is called the lytic cycle, where we have a few key steps that we'll talk about, and then we'll look at a handy picture of these in a little bit. So the first step is what we call attachment or adsorption, not absorption, but adsorption, where they target part of a bacteria or eukaryotic outer surface. Now we're just going to talk about bacteriophages because it's a bit simpler. So they target part of a bacterial outer surface, typically a protein, but they basically target it to basically attach. The next thing they do is penetrate or inject their host. So they basically pierce the cell wall and the membrane of the bacteria to inject the viral genome. Next step is synthesis, where the phage takes over the cell's replication and protein synthesis enzymes to synthesize brand new viral components. Then we have assembly. Now, once all those components are assembled, we have to put them together into new bacteriophages, and that's assembly. Then we have release, where mature virus particles are released through an enzyme that essentially lyses the host or buds through the host cell wall. And then finally, we have the eclipse period, whereas the time between adsorption and the formation of a new viral particles. And so if a cell is lysed at this point, few, if any, virons can be released. So that's kind of the flow. Attachment, you inject your genome. You use the genome to make new parts of the virus. You have to assemble those parts into new viruses, and then you have to release those viruses so they can then go on to infect brand new viruses. But again, it's kind of like it's like a step-by-step factory, which is kind of fun. Then we can look at the what we call the lysogenic fire cycle. Now, this is what we also call the latent phase, where the virus does not immediately kill an infective host. Now, the same original steps occur. There's attachment and then injection of the genome. Now, instead of assembling brand new parts of a virus, the virus integrates its nucleic acid into the host genome. Now, this integration allows the virus to be replicated along with the host DNA as the host divides. So basically, the virus just becomes part of the bacteria. When I say the virus, I just mean its genome. Its genome becomes part of the bacteria's genome, or your genome for that matter. This is the same process inside humans. Now, there are two distinct types of lysogenic cycles here. It can be temperate or lysogenic phages. Now, once the virus's genome is incorporated into the chromosome, it's what we call a prophage. So once the virus is part of the bacteria's chromosome, it's a prophage. And then a cell containing a prophage is called a lysogen. So let's talk about a phage, a bacteriophage, which is phage gamma. Phage lambda. Sorry, gamma would be if this was flipped vertically, but it's phage lambda. Now, this affects E. coli, and it's one of the best studied lysogenic phages on the planet. Now, when phage lambda infects a cell, the early events constitute a genetic switch that will determine whether or not the virus is lytic or lysogenic. Now, depending on the environment around it, the phage will basically become lytic or lysogenic. Now, it will essentially induction occurs basically during a period of stress. Now, the prophage can be excised and begins the lytic cycle, i.e. producing new copies of the phage, but it basically requires the churning of the gene expression necessary for the cycle. So let's look at just a visual breakdown, right? This is the lytic cycle, this is the lysogenic cycle. You'll notice that they're connected to one another. But we have that first part, attachment. The virus attaches to the bacterial cell wall. Penetration, you see the virus stabs through the bacteria, and it is exactly as violent as it looks. It just like pierces it like a harpoon, ejecting its DNA into the cell. Now, one of two things happens. We have synthesis, where we start to build new virus, and assembly, then release, and this process repeats over and over again. Or we have integration, where the virus's genome is integrated into the bacteria, forming a prophage. Then we have propagation, where the prophage is basically replicated along with the bacterial's genome. Now, sometimes this propagation can be indefinitely. So sometimes prophages hop into a genome and never come out. They just kind of are replicated indefinitely. And so we actually see lots of remnants in the human genome, where there's viruses that are really, really old that did this lysogenic cycle with us, but just never escaped. And they're just permanent parts of our genome. So kind of a fun thing. But eventually, if there's enough stress in the cell, there'll be a process called induction, where the prophage exits the bacterial chromosome, and then synthesis begins, followed by assembly, then release, and then the lytic cycle. Right? So there's this connection, right? The bacteria can be lytic, where it's producing new viruses, killing the host, or it can be lysogenic, where it essentially propagates itself through time. Now, we have this process called phage conversion. Now, during integrated portion of the lysogenic cycle, so this part here, Some viral genes may be expressed. Now, there are phenotypes or characteristics of a lysogenic bacterium that can actually be altered by the prophage. So, for instance, Vibrio cholera. Vibrio cholera is a normal, happy bacteria. It lives in the ocean. It likes to eat chitin, which so it means it likes to live on the exoskeletons of crabs and lobsters and things like that. However, Vibrio cholera, as you guys know, causes cholera, which is a pretty nasty gut infection. So if you guys remember from a few years ago, that big earthquake in Haiti that killed, was it 10, 15,000 people, and many of them died by cholera. Well, how do we go from happy cholera that likes to eat crabs to cholera that kills humans? Well, it's actually kind of cool. So cholera becomes bad cholera when a lysogenic phage introduces a gene that codes for a cholera toxin. Now, because it's lysogenic, the gene becomes incorporated into the host genome and converts a harmless bacteria into a disease-causing form. So if you've had any more reason to hate viruses after COVID, well, you can thank a virus from turning Vibrio cholera into a happy bacteria into a sad disease-causing bacteria. Um, Now, viruses, once they infect you, they can have a couple different avenues. Actually, before I move on, I just want to make mention this cycle. I know we talked about it in relationship to the bacteriophage. Are acute, and that's kind of like most diseases on the planet. But we categorize based upon two things. So the first being how rapidly and frequently the virus is produced, i.e., how many viruses is it producing every time a lytic cycle occurs, as well as in the appearance of associated systems. Now, persistent infections, they can be latent or chronic. So chronic meaning sort of going on for an extended period of time. They can also be acute, which is rapid replication of a virus and can lead to sudden symptom onset. And these acute infections, you can thinking things that cause outbreaks, epidemics, pandemics. You can think acute infection as in like COVID-19. You could think of something that's a persistent infection, something like HIV that basically stays in your body for a very extended period of time. So let's look at a couple of these. Now we have four distinct types of infection: acute, chronic, latent, and then slowly progressing. And you'll notice we have time on the x-axis and it's arbitrary, but you'll notice that we have, for acute, we have essentially infection basically is like the amount of viruses produced. You could think of it that way, or the severity of infection. But you notice that we have, um, this an acute infection, i.e. influenza, pretty heavy area of disease causing, but it doesn't last for a super long period of time. We could look at a chronic infection, again, lots of virus, and then it kind of hangs out for a really long time, and then it whips back up again. So this is what we see in hepatitis as well as HIV. Intense infection in the beginning, subsides for a bit, and then comes roaring back, um, roaring back, and as the virus continues to do its thing. Then we can have latent viruses, such as the herpes viruses, where they cause a disease, they die off, cause a disease, die off, cause a disease, die off. And I shouldn't say die off; they just go dormant. And so you can think of, herpes viruses. If you've ever had a cold sore, you have, say, a stressful event in your life. Say you have a really big exam, and you have a cold sore that pops up, and the goes crazy, and then it goes away, and then maybe you have another stressful time in your life, and the cold sore pops up again, and so on and so forth. So that's our latent viruses. And then we have slowly progressing agents, such as virons or potentially viruses that cause cancer, such as HPV. And you notice that they kind of just ramp up over time, and then they get much more disease causing as time goes on. So four distinct types of infections that we see with viruses, as well as things that look like viruses, such as prions, which we'll talk about at the very tail end of today's lecture. So let's talk about the flu. I was going to talk about COVID, but I think we've all had enough of COVID this year. So I'm going to talk about influenza because it's a pretty cool disease. Now, influenza has been around for a really long time. It is one of the most infectious diseases throughout human history, and there's been major pandemics spread throughout our history, with the most recent influenza pandemic at the largest scale, I should say, being the 1919, what we call the Spanish flu, which has seemed to infect about 1/3 of the world's population at the time, which was, I think, if I'm not mistaken, about 2 billion or so. and killed about 50 million or so people. So really, really dangerous flu, but at a time where medical technology wasn't so great. I would imagine that if COVID popped up back in the 1900s, when the Spanish flu popped up, it would be much more deadly. But I think our modern technology and sanitation and all that stuff has made COVID be slightly less deadly. Not slightly, I should say, significantly less deadly than it probably should have been. Now, flu viruses are enveloped on animal viruses, and there are three types based upon their capsid protein. Now, there's type A, which causes serious epidemics in humans. There's types B and C that causes mild human infections. So the 1919 Spanish flu was type A, and our average year-to-year Spanish flu, our Spanish flu, year-to-year flu is type B and C. Now, there are subtypes that differ in the protein spikes. Now, these protein spikes are two different types: hemagglutinin, or H, which aids in viral entry, and N aids in viral exit. So you might have heard of like H1N1 flu, for instance, like the most recent bird flu was H1N1. Well, H refers to hemagglutinin protein 1, N refers to neuraminidase protein 1. So you can classify flu strains based upon what types of hemagglutinin and neuraminidase that they have, as well as these type A, B, and C. Now, it's kind of an interesting thing. H and N molecules accumulate random mutation. The flu is an RNA virus, and as I mentioned, RNA viruses mutate relatively rapidly. Now, H and N molecules, so that hemagglutinin and neuraminidase, they accumulate random mutagens, which is what we call antigenic drift, which are essentially small changes in the HA and NA proteins, such that previous vaccine-induced immunity is no longer effective. So if you've ever wondered why you have to get a flu shot every year, well, it's because the flu from this year is going to be different from the flu from next year because of antigenic drift, i.e. changes in the HA and NA proteins. And you might ask yourself, well, why is that important? Well, our immune systems recognize different strains of the flu by recognizing different HA and NA structures and characteristics. So if it's changed enough, the immune system can no longer recognize it, well, you have to go out and get a brand new flu vaccine. Now, this antigenic drift has caused pandemics. So flu viruses that undergo genetic recombination when two strains infect the same cell. So if you have two flu strains, say one from a human and one from a pig, they infect the same cell at the same time. Well, the viral genomes are actually reassorted during this infection. And now this antigenic shift process of combining two distinct genomes together produces strains of the viruses with novel combinations of HA and NA proteins. So kind of a crazy thing, right? So when you hear like bird flu or swine flu or any other different types of flus out there, well, what's going on? Well, it's a virus that infects humans mixing with a virus that infects pigs, thus giving us our weird strains of flu. Now, as I've mentioned, flu is one of the most deadly things that we've ever seen. We talked about the Spanish flu. 50 to 100 million people worldwide were killed. This was H1N1, which is kind of dangerous. Now, the Asian flu of 1957 was H2N2. And it killed over 100,000 Americans. Hong Kong flu of 1968, H3N2, infected about 50 million people in the United States and killed about 70,000 of us. So again, flu, varying levels of deadliness, but again, all resulting from these antigenic shifts and recombination of different strains of the flu virus. So let's just look at this. I think it's a really cool system. So we can think of regular antigenic drift where we have a virus. Now, if we have a virus that's been infecting humans, say we got a vaccine to it, or has a potential to affect humans, and pretend there's a vaccine. Now, we've developed antibodies to it, and the antibodies target the H and N proteins on the cell surface, thus recognizing the flu. Now, if antigenic drift has occurred and thus a mutation has occurred, well, as you can see, we've changed the structure of the H and N proteins. So they're blue here, now they're red here. Thus, our immune system can no longer recognize it because it's essentially a brand new strain of the flu. Now, that's antigenic drift. Now let's look at recombination, where we have an avian influenza and a human influenza. where it has the HA and NA spikes. As you can see, they're distinct between the human and the bird version. So it's a pretty dapper looking duck, if I do say so myself. Now, if we pretend, and this happens quite a bit, as, as much as you would like to think it doesn't, it actually happens quite a bit because of how close we live to our livestock. But a virus from the bird and the humans infects a pig at the same time, thus mixing their genomes, thus producing novel combinations of HA and NA, being a partial combination of, say, our genome and the HA-NA proteins of our both avian counterparts and our human counterparts, creating a brand new novel virus, which I think is pretty cool. Right, this hybrid virus, similar group of viruses infect different hosts. So if I try to give this to the duck, it wouldn't infect the duck. If I try to give this to the human, it wouldn't infect the human. But with this recombination inside of a pig, well, now we have this what we would technically term as a swine flu. Now has a combination of birds and humans, thus can infect both birds and humans simultaneously. So kind of fun. So that's the flu. Let's talk a little bit about our newest pandemic, which is HIV, or the human immunodeficiency virus. Now, HIV is eventually causes AIDS. So latent stages of HIV infection give us acquired immunodeficiency syndrome, or AIDS. Now, HIV is a chronic infection, as we showed on that previous graph. Now, HIV infects and ultimately destroys certain types of immune cells. Now, the ones it likes to target are the CD4 positive cells, as well as our T helper cells and some classes of macrophages. So we have this virus that targets our immune system, which is why it's immunodeficiency, because it essentially kills off part of our immune system. Now, without CD4 positive cells and helper T cells, the body simply can't mount an effective immune response. Now, without our immune system, we won't inherently die, right? If we have no immune system, we won't die. However, if our immune system is damaged pretty heavily and we're say presented with a fungal pathogen or another virus or a bacterial pathogen, well, without an effective immune system, we can die from average everyday garden variety pathogen. So imagine you have a harmless version of the flu and you have a damaged immune system because of HIV. Well, that harmless version of the flu might actually kill you if your CD4 and T helper cells, or your helper T cells, I should say, are simply cannot mount an effective immune response. And people that eventually pass away from HIV usually get what we call opportunistic infections. So bacteria that normally don't cause the disease, but because their immune system is weakened, they essentially take advantage of the opportunity and infect the host. So let's talk about the life cycle of HIV, and it's a modern day pandemic. HIV has infected hundreds of millions of people, and has helped lead to the deaths of millions of people. So it is a modern day pandemic. It's a chronic pandemic, but it's a modern day pandemic nonetheless. Now, HIV attaches, so its attachment cycle, it only attacks those CD4 positive cells. Now, there's a protein called the viral GP120. Now, this attaches to CD4 proteins on macrophages and CD4 positive cells. So it's, again, targeting directly this one class of protein. Now, there are other co-receptors called CCR5 or CXCR4 that are also required. Now, fun fact, there's a group of people that have a mutation in their CCR5 gene, so the gene that encodes for this protein, that actually makes them functionally immune to most strains of HIV. So while CD4 is important, CCR5 and CXCR are also required processes. If you had a mutation here or here, that means the virus couldn't recognize it, you would actually be functionally immune to HIV. So once they attach, the viral enters through a process called endocytosis. And so basically it essentially, you can think of like it attaches and then essentially like melts inside to the cell. That's kind of a simplified way of saying endocytosis. Then we have replication. Now, HIV, as I've mentioned, is a retrovirus. So the enzyme reverse transcriptase converts the viral RNA to a double-stranded DNA. Now, reverse transcriptase is a naturally error-prone enzyme. So it ends in ASC, so it has to be an enzyme. It is really, really error-prone. It has an error rate of about 1 in 1,000, sometimes higher, sometimes less common, but the average is about 1 in 1,000. And so mutations in HIV are super duper common, like super duper common. So once reverse transcriptase converts the RNA to DNA, the DNA is then incorporated into the host genome, and there's a variable period of dormancy. So before the HIV really ramps up and starts, building um, copies of the virus, there is a period of dormancy. Then we have assembly and release, so making new copies of the viruses, then releasing the viruses, and they exit via budding. So let's look at this in a physical 3D sense. So we have the the GP120 glycoprotein on the surface of the HIV envelope. HIV is an envelope virus. It binds to CD4 and one of the other co-receptors on a CD4 cell. The viral contents then enter the cell by endocytosis. Again, it's like kind of a melting of the viral parts and allowing the virus to come into the cell. Then once the virus's genome is inside the cell, reverse transcriptase catalyzes the synthesis of DNA from RNA, and that DNA is then incorporated into the host's own chromosomes. Now, once it's in the host chromosomes, regular transcription processes mediated by the host are done. So standard central dogma of biology, DNA to RNA to protein. Now, once we produce all the different parts of the viral protein, we have to assemble them into a virus. So you can see the viral capsid surrounding by the viral DNA, and then the virus exits by budding. And so you'll notice that the virus has attached its own proteins, so those GP120 glycoproteins. But again, this envelope, it's just part of the host. It's taking a part of the host with it. And as a matter of fact, if you were wondering this, envelope viruses are really hard to get rid of because it's hard to distinguish them from the host, right? Because they're part host, part virus at the same time. So that's HIV. That's how HIV infects. But again, you can see the standard cycle. Of the error-prone nature of their reverse transcriptase. So during an infection, HIV is constantly replicating and mutating. Now, eventually the virus mutates enough to eventually viruses that mutate enough, they can infect a broader range of cells, leading to the loss of T helper cells. So they start on those CD4 plus cells. With enough mutations, they can then infect T helper cells. And the destruction of these T cells blocks the body's immune response. Thus, cancers and opportunistic pathogens can occur much more readily. So that's HIV. Now, how do we treat HIV? Well, there's a couple ways. Now there's many medications out there that have propped up in the past 30 or so years. So there's a couple of them. So there's enzymes that block maturation. They're called protease inhibitors. There's those that block integration. They're called integrase inhibitors. There's those that block replication, which are reverse transcriptase inhibitors, NRTI and NNRTIs, just as a note. There's those that block entry and those that block attachment. So there's any parts of that lytic cycle we can potentially target with medication, but not all medications are effective for all strains of HIV, and they're not always effective on different hosts. Now remember, HIV uses lots of the host's machinery, so not every host responds, and I say host, I should say every human responds well to many of these drugs. So maybe this works for one person, it might not work for another person. So HIV has kind of a wide variety of treatments. So, and the other thing I want to make mention about HIV has a high mutation rate. So if you got infected with HIV, you actually, with enough time, actually develop your own unique strain of HIV that has multiple substrains with it. So your strain of HIV, if you happen to get it, which I hope no one ever does, but it would be distinct for you given enough time relative to, say, another person in the population that has it. Even say if you compared your HIV to the person who gave you HIV, your HIV would be distinct at the genomic level, of course. So fun stuff. So let's talk about latent viral infections. Now, these are viruses that hide in the host to invade immune system, and they can actually establish lifelong latent infections. So herpes simplex virus 1 and herpes simplex virus 2, they trigger acute disease episodes and then become latent inside neurons. And physiological stimuli like stress can trigger their reactivation and lead to additional disease episodes. So the example I gave you earlier was, you say you have a cold sore, which is caused by H. herpes simplex 2, if I'm not mistaken. You get stressed, the cold sore flares up, right? That's the infection, then dies off, and it hides in your neurons. So it's kind of a jerk like that. But that's what our latent viral infections look like. Now, as I've mentioned, viruses can cause cancer. Now, one of the best studied viruses that causes cancer are the human papillomaviruses, or HPV. And about 70% of all cervical cancer cases are caused by the sexually transmitted HPV virus. So if you have not got the vaccine for HPV, you should definitely do it. Even if you're a guy, you can still get it. It works for both male and females, thankfully. Now, viruses cause cancer by altering the growth properties of human cells, and they do this by injecting their genome. Now, the genome can change the behavior of the cell, and it does this by potentially blocking cell death and promoting cell division. And that's kind of what's going on, right? The virus is altering the behavior from a normal cell to a type of cell that resembles a cancerous cell. And cancer cells simply don't die, and they have uncontrolled cellular division. So viruses can change the behavior of cells just by infecting you. And again, HPV vaccines work great and they help prevent cervical cancer. So if you haven't got one, definitely get one. If you have a partner, you should definitely ask them to get it. If of course they're willing to, because they work really well to prevent HPV infections, which again can cause cancer. And again, cancer is pretty obviously a very serious thing. And so the final thing I actually want to make mention is of our prions, which are proteinaceous infectious particles, or prions for short. Now, prions are wild. They really are. And you'll notice from their name, it's proteinaceous. So they're proteins that have gone essentially to the dark side and now cause infections, which is wild, right? Viruses are DNA or DNA or RNA and protein. Prions are just the protein part. Now, the most common, or at least as far as we know, all prions cause what we call transmissible spongiform encephalopathy. That word, TSE. Sorry, my pronunciation apparently is not very good today. Examples of these include mad cow disease, or BSE, scrapie in sheep, wasting disease in deer, as well as Creutzfeldt-Jakob disease in humans, or CJD. Now, the host has normal prion proteins, which from a genetic capacity is labeled as PRP superscript C. Now, all the prion is we take this PRPC and we misfold it. Now, you might remember from biology 1 that the protein structure, i.e. how the protein folds, dictates its function. And if you misfold a protein, or if you have a mutation that causes the protein to change shape, well, that changes the protein's function. And that's all that's happened with a prion. So misfolded proteins are just prions. And we denote these as PRPSC. Now, misfolded proteins are actually kind of funky. Now, these cause these are the prions, right? These are the causing the disease. But the interesting thing is if a PRPSC encounters our PRPC, it can actually convert the PRPC into a PRPSC, thus making it a prion. So normal PRP protein, as is necessary for the productive infection by scrapie as a molecule. So if we have a mouse that's infected with PRPC, thus it has scrapie. If you take its brain homogenate, and let me tell you what that is, you just grind the brain up in a blender. Now, if you have a mouse that has a PRPC null mouse, i.e., it's basically has a mutant, and it can't be converted into PRPSC, the mice will actually live a normal lifespan and the brain appears normal. So if this mouse basically has a slight genetic variability to it, it can't be infected by prions, this type of prion, of course. Now, those that are PRPC wild type, well, they die of those neurological disorders, the brain prion. So funny group of organisms. I think really scary as a pathogenic agent, and they're really brand new sort of area of biology that we just don't quite fully understand. If anything I told you about these prions got you excited, I definitely recommend doing some research on them. There's simply not enough time in this class to talk about prions, but they are really wild organisms. Um, and the final thing I want to make mention is this idea of a viroid. Now, viroids are tiny naked molecules of circular RNA. So we went from viruses, which are proteins surrounding DNA or RNA, to prions, which are just a protein, bad proteins. And now we have viroids, which are just tiny naked molecules of RNA. Now, these cause disease, which is why we're talking about them, which is again weird. As far as we know, they don't infect humans, but they do cause disease in many agriculturally and economically important crop systems. But basically, what they do is they use the host protein to replicate, and they cause the host to produce small interfering RNAs or siRNAs during the infection, and these interfere with plant growth and development. So again, wild areas. Not we know this course is just a broad survey of life as a relates to diversity. So we don't get to dive too deep into these, but I definitely recommend if you have a few moments, Google some information about prions, Google some information about viroids, so you can learn about these wacky and wild areas of biology. So with that, that is going to be the end of today's class. I do hope you learned something. And if you have any questions, please feel free to reach out to me. But otherwise, I hope you guys have a great day. Take care.